2, 3. Alright, good evening to all of the members of the floor today. We're going to present our research proposal poster, which is titled A Simulation of Piperine from Sarawak Black Pepper for Anti-Inflammatory Activity using LPA-stimulated Rho267.7 macrophages cells. Today, there are four of us who will be presenting about this poster, which is the first one is that me, Marren, after that Aisha Claire, DAMIA SYAFIERA, and LESTON MANJA. Without wasting any more time, let us go straight to the abstract today that will be presented by Miss DAMIA SYAFIERA.
Good evening, I'm Damia and I will explain about the abstract. Piperine, the principal alkaloid of black pepper or Piper nigrum, exhibit a broad range of therapeutic effects including anti-inflammatory, anti-cancer, antioxidant, and neuroprotective activities. It also enhances the bioavailability of various drugs and phytochemicals. This review compiles recent clinical and preclinical evidence on piperine's molecular mechanism, pharmacokinetic, nanoformulations, and its potential in disease management. Quantification was performed using optimized extraction techniques followed by high-performance liquid chromatography. Anti-inflammatory activity was assessed through nitric oxide production and inflammatory marker reduction. Findings support the efficacy of piperine in attenuating inflammation, highlighting its promise as a natural candidate for anti-inflammatory drug development. Thank you.
Alright, thank you Ms. Damia Shafira for the presentations about abstract. Now we'll be moving on to the relative diagram that we found out from the research that we have reviewed. Alright, we have reviewed an article from Tripathi et al. And this is the diagram that we found out. This diagram will be presenting about the molecular targets of piperine. We could see that there are three crucial components about inflammatory, which is AP1 and FKB and COX-2. AP1 and FKB are the transcription factors. They will be sending signals toward the inflammation enzymes, which is COX-2, and FKB and AP1 works by inducing the genes by translocating themselves to the nucleus and induce expression of pro-inflammatory cytokinase and other mediators. Meanwhile, enzymes such as COX-2 will induce prostaglandin, which is the potent mediator for inflammatory, and this mediator will be able to induce whether inflammatory action to happen or not. Okay, so with the excessive production of prostaglandin due to the production of COX-2, this will also allow other metabolic diseases to occur, such as tumor and cancer. But with the help of piperine, we could regulate these diseases. This also shows that piperine not only acts as the inflammatory reducing agent, but it also will be available for other therapeutical agents towards other diseases. This also opens up the way to provide that piperine will have its potential towards other medicinal, pharmaceutical drugs or medicine. Now let's move on to the result that were presented by Ms. Asha Claire.
Okay, I'm going to explain on the result part of this article. To date, at least there are 11 completed and 5 ongoing clinical trials have been evaluated the therapeutic potential of piperine in a range of human health conditions. These include metabolic disorders such as diabetes, infectious disease like AIDS, neurological disorders including epilepsy, and also inflammatory diseases such as osteoarthritis. The trials vary in scale and design, but collectively, they demonstrate a growing clinical interest in piperine's pharmacological effects and safety profile. Next, piperine exhibit favorable pharmacokinetic properties, notably its ability to cross the blood-brain barrier, making it a promising candidate for neurological applications. Furthermore, piperine significantly enhances the bioavailability of co-administered compounds such as curcumin and paclitaxel by improving intestinal absorption. This bioenhancing effect is of particular importance in drug formulation and delivery systems. Lastly, Mechanically, piperine acts on multiple molecular pathways and targets. It modulates inflammatory and cell survival pathway, including NFKB and all of the listed pathway in this poster. Additionally, it influences the expression and activity of various enzymes, receptors, and transcription factors involved in inflammation, carcinogenesis, and also neuroprotection. This multi-target action support its potential as a therapeutic agent across diverse disease states. Thank you.
Thank you, Ms. Asha, for the clarifications about the results. Now we're moving on to the interesting part, which is discussion that will be presented by Mr. LESTON MANJA.
So, hi, I am Leston. So, now I'll explain the key discussion points of our study. Piperine, as shown in our findings, supported by the review article by Tripathi, works mainly by inhibiting NFKB pathway, which is a regulator of inflammation in the body. When an FKB is blocked, it reduces the expression of several pro-inflammatory proteins, especially the tumor necrosis factor alpha, TNF alpha, and inducible nitric oxide synthase, INOS. INOS is responsible for producing nitric oxide, which is a signaling molecule involved in the inflammation. In our study, we observed that piperine treatment reduced nitric oxide levels in macrophages, indicating that the inflammatory response was successfully suppressed. This effect reflects a direct disruption of inflammatory signaling, confirming piperine's anti-inflammatory mechanism at cellular level. These findings are consistent with what has been reported in the review by Tripathi et al., which also discusses piperine's impact on various inflammatory pathways, including the NFKB, COX-2, and the cytokine regulation. So our results are strongly aligned with this existing scientific literature. What makes our work unique is that the use of piperine extracted from the Sarawak black pepper, a botanical source. This is not only to demonstrate its biological effectiveness, but also its regional significance, showing that a widely available local spice has a real pharmacological value. The potential application here is quite meaningful. Piperine could be developed into plant-based anti-inflammatory drugs that may offer fewer side effects compared to the compared to the long-term use of synthetic drugs like in NSAIDs. It could be also a complementary therapy, enhancing the action of conventional treatments and possibly reducing the acquired doses. Beyond health benefits, this research also supports the utilization of local agriculture products, such as the Sarawak black pepper, in medical innovation. This aligns with the efforts to promote sustainable pharmaceutical development and could help drive local economic value through verbal product industry. And lastly, to move toward clinical use, the next steps is to include in vivo studies and formulation improvement such as nano formulation to improve absorption, delivery, and therapeutic effect of piperine in the human body. That's all. Thank you.
All right, thank you, Mr. Leston, for the really wonderful presentation about the discussion part. Now we're moving to the last part of our presentation today. First, we would like to conclude that piperine is a multifunctional molecule with the proven therapeutic potential in inflammation such as cancer, neurodegeneration, and metabolic diseases. This is proven by its capability to control a lot of molecules in the body through the signaling pathway control. It also improved drug delivery and treatment efficacy, which has been further discussed in the discussion part just now. And it also provides a continued translational and clinical research which will validate its broader application in human health, and it also boosts the popularity and the usage of Sarawak black pepper. So now we're moving to the acknowledgement that I would like to give an acknowledgement to Tripathi et al. For providing such a very informational article that is being used for our article today, and let us also not to forget giving acknowledgement to our scientific communication courses lecturer, which is Madam Safarina. So now we have just moved on to the final part of our presentation, which also marked the end of our presentation today. Thank you everyone for lending your ears and eye to watch and listen our presentation today. Let's hope that we meet once again in the future. Thank you everyone.